Hello, my name is James Lamp, and I'm a research technician working in Dr. Vincent Setola's lab. Today, I'm going to talk briefly on some of the work that we are doing aimed at discovering new strategies to prevent and/or treat substance use disorders. Our research is aimed to better understand the brain cells and neural circuits that underlie behaviors associated with substance use disorder. Simply, all drugs of abuse increase dopaminergic neurotransmission in a part of the basal forebrain called the nucleus accumbens. Dopamine neurotransmission in the nucleus accumbens is typically associated with pleasure from rewarding activities such as eating and socializing in a well-controlled manner. Drugs of abuse, however, hijack this dopaminergic mechanism in the nucleus accumbens, which leads to substance use disorder-associated behaviors. These dopaminergic neurons in the nucleus accumbens are innervated from a midbrain nucleus called the ventral tegmental area, or VTA. Our lab focuses on the neurons between the VTA and the nucleus accumbens and how to effectively modulate them in order to prevent and/or treat substance use disorders. Our lab focuses on the neurons between the VTA and the nucleus accumbens and how to effectively modulate them in order to prevent and/or treat substance use disorders. Our lab has three primary questions. First, what G-protein coupled receptors, or GPCRs, are expressed by dopaminergic neurons between the VTA and the nucleus accumbens? GPCRs are the most prevalent and pharmacologically targeted receptors found in the nervous system. Knowing precisely which GPCRs are expressed by VTA to nucleus accumbens dopaminergic neurons would enable us to identify already existing, FDA-approved drugs that could decrease or suppress dopamine release in the nucleus accumbens. This could either prevent the onset or treat substance use disorders. Second, we wish to discover what brain regions project to the VTA and modulate the activity of the VTA to nucleus accumbens dopaminergic neurons. Just as the VTA influences the activities of the nucleus accumbens, brain regions upstream of the VTA can have an indirect pathway to suppressing substance use disorder-associated dopaminergic release. To do this, we use a technique called retrograde tracing. We inject the fluorescently labeled virus into the neuron that we wish to study. This highlights the entire neuron a color that we can image and study its location. Here I have an example of what we would see if we did this to the nucleus accumbens. The VTA is lit up with color and is clear to see versus the rest of the brain. Now, here's an example of what we might see when this is performed against the VTA. These fluorescent regions can further be isolated and formed into a single cell suspension, which allows us to use transcriptomic techniques to identify all the various GPCRs or GPCRome found within these highlighted regions. This GPCRome can presumably provide many more treatments that can be utilized to prevent and/or treat substance use disorder in addition to the targets already known on the VTA. Finally, we wish to see if we can leverage our understanding of the GPCRome of the VTA to nucleus accumbens dopaminergic neurons and of the neurons that project to and influence the VTA to prevent and/or treat substance use disorder-like effects in mice. Many drugs of abuse that are rewarding to humans also exhibit rewarding properties in mice. For instance, mice given free access to self-administered intravenous infusions of oxycodone or cocaine will consume increasing amounts of drug over multiple sessions in an assay known as intravenous self-administration, or IVSA. During abstinence, i.e. removal of the drug, those mice will engage in a behavior reminiscent of compulsive drug seeking in humans. Mice that successfully unlearn drug self-administration after a period of abstinence will, under exposure to stress or drug-related cues, reinstate drug self-administration behavior. Thus, we can model acquisition and escalation of drug-seeking behavior, drug withdrawal-associated behaviors, and relapse of drug taking following a prolonged period of abstinence using mice. We are leveraging this ability to test for existing and new pharmaceuticals that may have clinical utility for preventing and/or treating substance use disorder.